Hi, this is Dr. Robert Norman. Glad you're here. This is the Brain-Skin Connection series, The Outer Brain, the Incredible Power of the Brain-Skin Connection. This is from the Norman Center for Neurodermatology. We're investigating the interactions between the brain and the skin in relationship to disorders such as melanoma, Alzheimer's disease, Parkinson's disease, autism, neurodermatitis, and other neurocutaneous diseases. What is our goal here is to translate skin-brain research into effective behavioral strategies that prevent disease, promote healing, provide treatments, and enhance well-being across the lifespan. How does the human skin microbiome influence our brain and mental status? Our skin reflects critical changes to the way we think, feel, and perceive. We are learning new ways in which our skin microbiome influences our perception and cognition. This is an overview slide of the normal microbiota of the skin. And here we see Staphylococcus, Micrococcus, Diphtheroids, and Malassezia furfur. These, again, are normal microbiota. When they change in amount and content and other issues, it can cause problems and disrupt the skin and disrupt our quality of life. The skin is the largest organ of the human body and is colonized by beneficial microorganisms, as shown in the last slide. The skin microbiota is composed of millions of bacteria, fungi, and viruses. Key roles in the skin microbiota, in similar fashion to the gut microbiota, is to educate our innate and adaptive immune system, protect against invading pathogens, and break down natural products. Studies of the human microbiome have revealed that even healthy individuals differ remarkably in the microbes that occupy habitats such as the gut, skin, and vagina. Much of this diversity remains unexplained, although diet, environment, host genetics, and early microbial exposure have all been implicated. Accordingly, to characterize the ecology of human-associated microbial communities, the Human Microbiome Project via the NIH has analyzed the largest cohort and set of distinct, clinically relevant body parts so far, and that is noted, body habitats so far, and that is noted in this this link right here. You can get to their studies. Here is a topographical distribution of bacteria on various skin sites, which obviously took a tremendous amount of research, but it actually shows in which body parts the more common bacteria and other species are located. The skin is colonized by diverse microbiota that is adapted to utilize the nutrients available on the skin. Although there are myriad environmental perturbations over time, the skin microbiota of a healthy adult remains mostly stable. Altered microbial states can create or alter skin diseases. How do the gut and skin microbiota differ? Microbial communities in the gut stabilize around 3 years of age, with most strains likely acquired from family members and close contacts. Initial colonization of the newborn skin depends on the way the baby first enters the world. Vaginal delivery allows bacteria colonization with a vagina, but with cesarean section, the babies acquire skin microorganisms first. Skin microbial species change during puberty due to a rise in hormones that fire up the sebaceous glands to produce more sebum. These changes result in marked variations in the bacterial and fungal landscape and predict which skin diseases will be more likely compared to the pre-pubertal period. The gut is filled with all kinds of microbiota-friendly juicy meals as compared to the much less diversified and robust diet provided to the skin microbiota. For the skin microbiota, the issue of survival depends on a menu selection of basic proteins and lipids in an acidic, cool, and desiccated environment of sebum, sweat, and the stratum corneum. Much has been written lately about the gut microbiome and the brain. We have many unanswered questions. How does the human skin microbiome influence our brain and mental status? What is the crosstalk between the immune system, the brain, the mouth, the gut, and the skin microbiota? Studies have shown that our microbiome may play a role in mental health and neurological conditions such as autism, epilepsy, and depression by interacting with our nervous system and even releasing molecules that can perhaps make their way to the brain. More studies certainly need to be done specifically on skin microbiome and brain function. What about the effect of geographical location, example China versus United States, on the microbiome? If the skin microbiome affects the brain, could different species inherent in different geographic regions affect brain function differently? In my next lecture, I will discuss how can the skin provide a way to detect brain abnormalities.